Hello and welcome to this webinar titled Current and Future Applications of Near-Infrared in Pharmaceutical and Biopharmaceutical Industry, hosted by Biopharma Asia magazine and presented by Dr. Erik Skibsted and Dr. Fabien Chauchard. My name is Sri Patel and I will be the moderator. Before we begin, I would just like to remind our viewers that this event will be hosting a live question and answer session at the end of the presentation. You can submit a question at any time throughout the presentation via the Questions tab located at the bottom of your screen, and we will go through these at the end of the presentation. Now please allow me to introduce you to our presenters. Dr. Erik Skibsted has a master's degree in chemistry from the Technical University of Denmark. He received his PhD degree from the University of Amsterdam, specializing in near-infrared spectroscopy, multivariate data analysis, and solid dosage form manufacturing in the Process Analysis and Chemometrics group under the leadership of Professor Ag Smoller. He has 17 years of experience from pharmaceutical research and development at Novo Nordisk. Dr. Fabien Chauchard started working in the aeronautical and semiconductor industry as a process engineer. After a few years, he started a PhD at the research center RCPA French Research Center. During his work, he collaborated with Lund Laser Center in Sweden to develop new optical techniques to analyze solid sample and turbid liquid. At the end of the thesis, he founded with Dr. S. Russo Inkjet Company in order to bring new SRS SAM-Spec technology on the PAT pharma market. I would now like to invite our first presenter, Dr. Erik Skibsted, to join our webinar. Welcome, Dr. Skibsted.
Thank you, Sri, and good morning to all of you. First, I would like to say that if you have some questions which you will come up with later, you can contact me on the email or send me a text on this you see on this slide here. So what I will talk about are some trends and opportunities which I believe would cause that we use much more near-infrared in the pharmaceutical and biopharmaceutical industry. Near has been used for many years for raw material identification, quantification of residual water in lyophilized drug products, as inline technology for fluid bed dryers and in powder blending processes, and for assaying tablets. But there will be some more applications coming up in the future, and I hope to give you some insight into that. First of all, the rise of personalized medicine. I will give you a little bit comment on that and how near-infrared can support that. I will talk more in detail about biopharmaceutical manufacturing efficiency with some work I've done recently. And then there's a big trend called continuous manufacturing, where near-infrared will play a very important role as well. Improved sampling, how you can improve sampling with near-infrared will give you opportunities to measure new applications with near. And last, I would give some examples from improvements in the regulatory guidelines, which also will support more near-infrared spectroscopy. So, about the personalized medicine, the whole idea of personalized medicine is that you take into account each person's unique genetic, genomic, or clinical or environmental information, and then you give them a drug that is personalized for the patient. One of the quality parameters that is probably will be most personalized would be the dosage, the strength of the drug product. So I believe that would be the first quality parameter that is individualized. Depending on how the person, how big the person is, the race, kidney and liver function, there can be a very huge difference in what dosage that is required for therapeutic effect. One example is Varifin. It's a blood thinner. There's a 20-fold difference in how to achieve a therapeutic effect of that drug. So as you see in this slide here, each person has very individual needs, and dosage could be one parameter that should be customized and personalized. So in the future, we might have a situation where each patient is diagnosed, and based on how the genetics, genomics, or the size of the patients, and they would get a individualized medicine. And there are some technologies that can support that. One is 3D printing, and the other one is inkjet printing. So there has been a lot of papers the last couple of years how these technologies can be used to produce drug products with great flexibility. And NearInfrared is one technology to support this movement here. In a paper by Vakili from 2015, they demonstrated how you could use a commercial inkjet printer to print on eatable paper a drug product, an asthma drug. They printed the area of 1 square centimeter and with different number of layers of print on top of it. And then they used near-infrared chemical imaging to assay the final drug product. You see here a picture where the color is after chemometric evaluation of the near-infrared data, and the strength of the red color is correlated to the amount of drug product that is deposited on each of these 1 by 1 square centimeters. So the plot to the upper right is a calibration plot that shows how well the near-infrared method is calibrated to a reference method that was HPLC. So this is actually a technology, near-infrared chemical imaging, that can be put into line after an inkjet printer. And these technologies are easy to handle and use. So it would be a viable methodology how to produce a drug product, for example, at the pharmacy, and be very close to the patient here. And I believe also such new technologies like inkjet printing and 3D printing can help if you have a product with very poor stability, because the time from production to the use of the drug can be very short, like in half an hour, something like that. So you don't need to have a drug that needs to be stable for 2 years as maybe a traditional stability requirement would be. So, and near-infrared chemical imaging, or near-infrared in general, is a very easy to use technology for quality control of the doses. Another example of trends which could support more use of near-infrared is the need for having a very efficient cell cultivations or fermentation processes. These processes are very complex, and you use very complex raw materials for that. For example, soy hydrolysis, and it gives a good value for money to use the right quality of the soy, because the product here is a high value. So a few percentage more from these processes would be worth millions of dollars in many cases. Near-infrared is not a specific methodology, but it has some qualities when it comes to characterizing a raw material as well, and I will demonstrate that in the next couple of slides here. So here's the process we have investigated. We use soy hydrolysis as a feed medium, and we use it for cultivation processes. And what we have experienced is that when we change to a different batch of soy medium, sometimes the yield goes very up or it goes down. But the problem is to analyze what component is inside the soy which causes such a change of yield. So in this study, we looked on different quality parameters here, or we counted the number of cells in the cultivation, the viability, the size of the cells, and the titers of this actual product. And we also looked at metabolites inside and see how changing qualities of soil would influence those parameters. So here's an example when we use 2 different soy batches for the same cultivation process in large scale here. So you can see the upper curve, the blue curve, that's a concentration of the titer measured during a 21-day period. And the green curve underneath is the same process, but we use different batch of the soy for the feed medium. And if you compare the area under the curve, this is a good estimate of how much product you will get out from this cultivation, there's a 2.15 ratio difference between that. So that's actually a very severe difference in the productivity when we just change the batch of the soil. If you look in the literature on soil hydrolysis and cell cultivations, you would see that people have used many technologies how to analyze the soil and maybe identify what is causing high or low productivity. You could choose many technologies like very specific technologies like NMR or LC-MS or something very advanced. And what you would come up with is that you have a raw material that has more than 1,000 individual components. And you would find, if you do a statistical analysis comparing the composition to the yield, that a lot of the components would cause an increase and some would cause a decrease in yield. NearInfrared is another technology that has been demonstrated. It's not specific, but because soy is amino acids and carbohydrates for most of the parts, very non-specific, Near is a very good candidate to analyze that, but as a gross composition method. The problem is you have very small spectral differences between a high and a low yielding soy. And I tried in the beginning to use regression modeling between the near spectra and the productivity, but it failed because I could not use the same predict model on small scale as in production scale. So, and the reference cultivations are nonlinear also. So what we have worked with is using classification model, and I will demonstrate that. So the way we build a classification model is that we take a lot of different soy batches, and then we do cultivations in very small research scale in an AMBER 15 microscale reactor. They are only 15 milliliters. We measure a lot of cell cultivation data, and then we analyze those data using principal component analysis modeling. And based on those models, we identify some soy batches we call good performing and some we call poor performing. So it's a binary classification here. We also measure the same soil with NearInfrared, and we build a classification model using PLS discriminant analysis. So the way we want to use that now is that when we get a new soil, we measure by Near. We use the spectra in the classification model, and then we decide, do we want to use that soil? Do we not want to use it, or maybe even mix it? So here's the AMBER setup. So for those of you not familiar with cell cultivation, it's a small 15 milliliters reactors that can be run automatic by a robotic system. And we run for 18 days, and we take our samples and measure for different cell cultivation parameters. For example, the titer, which is the product. Then we take all the data and we analyze with a PCA model. And the reason why we use all the data is because they all carry information about the variability caused by the soil. So in this study, we did 9 AMBER experiments. Each AMBER experiment is 24 small cultivations, so a lot of small cultivations here. And we tested 44 different soy batches, and we also did some mixtures. We used those data to create a soy performance label and did the calibration of the PLS-DA model. Some of the soy batches we also have used on laboratory scale cultivation and on full scale. And we verified that the performance of the soy batches were equally good or bad on a different scale to be sure we have a scale-independent methodology. So here you see the Near setup. It's a standard laboratory NearInfrared spectrometer, and we get from each soy batch 5 samples. We put in a vial and put a rubber stopper on and an aluminum cap. And by that we create a sample library we can analyze again and again to see if the instrument is stable and if there's a drift in the methodology, etc. So I use a fairly high spectral resolution of 4 reciproc centimeter because I was interested if there were small parts of the soil spectra that was relevant for this discrimination model. So in this slide you see spectra from 2 soil batches, 5 spectra from a good batch, and 5 spectra from a soil that is labeled as a poor batch. I needed to do some pre-processing before I could get out the relevant information, and I did first derivative pre-processing here, first derivative spectra. And here are two spectral regions that carry the most relevant information for that. You can see the green spectra, they are from the soil beds that we label as a good performing soil beds, and the blue spectra is from a soil beds we label as a poor performing soil beds. So you could start to interpret what are the main components in those two spectral regions, and then give feedback to the manufacturer of the soil and say how they maybe could improve their manufacturing process, so you only would receive good performing soil. This is the pre-processing I use for the PLS-DA model. And the next slide, you see the result of the PLS-DA model. The figure to the left is the classification error based on the number of latent variables. And I picked only one latent variable for this. It gave a 5% classification error. I tried to use more. You can see if you go to 9 latent variables, you would get 0 classification error, but that was clearly an overfit of the model. To the right, you see how the model samples of some good and poor performing soil batches were placed in the hoteling and the Q residual plot. I allow a few samples from the soil batch C01 to be above the 95% confidence limit. That's okay with me to give a little slack. It's a 95% confidence interval, so you would expect a few of the samples to be outside of that limit. So here you see from the PLS-DA model to the left is the regression vector. It shows what are the spectral regions where you have the information to classify the soil in good and poor. And to the plot to the right, you see the VIP scores. So it tells you which of these spectral regions are really important to classify soil in a good and a poor. And you see some dominant features around 4,500 and 4,200, around 5,000, 6,000 and 7,000 reciprocal centimeters. So you could elaborate more on the VIP score plot and see what are the chemical components that absorbs in those regions to learn about maybe some metabolism that is relevant for your cells. So in this plot is the plot to the right is the classification of the samples of the soil samples in a good classification, and you see the 5% error comes from the soil batch G13. They have a blue triangle just above the red line, that is a threshold. So they were misclassified as good performing, even though the study in amber showed it was a poor performing. But I would say this is okay. It's 5% is acceptable. And if you look to the left on the receiver operating characteristic curve, the ROC curve, you want a curve that is goes all the way up along the sensitivity, green y-axis, and to the horizontal on the specificity curve. So I was satisfied with this model here. So how can we use this? We use this model to look on a lot of soil batches used in full scale. We have a lot of historical cultivations with many different soil batches, and all these soil batches were analyzed with this classification model. A few of them were also used for the model, but most of them were not part of the model. So the red symbols here are the soil batches we have used historically in full scale. And the green and the blue are the calibration samples. So how can we use this? This is a classification of poor. So the way we could say, what about the soil samples, the soil batches that has been classified as poor? They are marked with the red square here. If we look how these soil batches performed historically, we will see in the next slide. This is this one. So these are actually tighter results measured historically in 48 full-scale cultivations. And all the red curves are the tighter we have experienced when we used the soil with the NearInfrared model classified as poor. So you can see historically, if we had the NearInfrared model many years ago, we could have avoided these batches, and they would give a very low yield compared to what we could achieve. So we want to use the NearInfrared method now to make better selections, so we would avoid in the future to have cultivation that gives such a low yield as shown here with the red curves. And I believe that NearInfrared has a big potential to look into other complex raw materials, also yeast extract, and some complex defined media also used in the biopharmaceutical industry. In other studies I have not shown here today, we have looked into what is called a defined medium with many components that we get a mixture from a supplier, and we could see batch differences, which also seem to correlate with some yield differences when you use it. So even though you end up with a defined medium, you need to put some quality testing on top of it to be sure to have the best performance processes. Another trend in the pharmaceutical industry is the rise of continuous manufacturing of tablets. The picture here is a continuous powder mixture, and this technology, continuous manufacturing, really is making a big revolution in the pharmaceutical industry these years. Traditionally, when we're manufacturing tablets, we use technology that is 50 years old, and it's made of small individual batch processes or semi-batch processes. Some of them are continuous to some extent. But what we do is we put one production equipment in one room, and then we produce a small intermediate product, and then we take this in a drum to the next room and put it into the next unit operation. It's a very inefficient way of producing. It requires a very big factory to do this, and it gives a very inefficient way of producing drug products. But the new way of doing it is to merge all these unit operations very closely together, and there are some very clever engineering coming out this year where you can squeeze the footprint of a tablet factory very, very big. Or these are some of the numbers that's been reported, like reduce the footprint of the factory with 64%, with half the manufacturing cost, speed up the development time with a very high factor here, reducing it 90%. You reduce the amount of API you need for process development, and the cost per each tablet is also reduced. This paradigm shift has been embraced by many of the very big pharmaceutical companies, and it's certainly something that is will make a change also the way we use NearInfrared, because controlling such a process is very important. It's a key to be able to do this. And NearInfrared is a very suited technology for monitoring many of these steps here, ensure that the production is running smoothly and on target. Here's a picture from Janssen Pharmaceutical, and the yellow circles with numbers in points to places during this continuous manufacturing line where you have inline technology, and NearInfrared is very suitable for measuring, for example, the moisture and the blend homogeneity after a continuous mixture, and moisture after a drying step, for example. So I will talk a little bit about how sampling has improved, or some tricks you can do to use NearInfrared for very difficult application. There are three things I would point your attention to is the use of spectral filtering, advanced sample interfacing, and advanced probe design. And I know Fabien will later on talk about SIS technology, so I'll not spend time on that today in my presentation. So here's an example of how we use spectral filtering to use NearInfrared in a very difficult application. We had a small probe inserted inside a tablet coder here. Very small batch amounts, sometimes there were only 2 or 3 tablets thick of the tablet bed. So here's the problem. We have a probe inside a moving bed of tablets. Sometimes you will analyze between the samples, between tablets. So that would cause a baseline shift. So how do we control that we only use data from when we put the light on the tablets? So here you see a situation to the left, how the data would look like if we just continuously scan during a coding process. And to the right, we have used spectral filtering. It's very easy to do and something you can try yourself. We use a Fourier transform instrument here, and we continuously collect scans, and then we sit and we average a number of scans to get a final spectrum. We set inside the software a threshold to look on the energy intensity of each scan, and if the energy is very low, it means that you receive light from in between the tablets, and you would discard that scan. And you would use the threshold to select the scans that have high energy intensity, and that would be the situation where you will receive information from the tablet surface. So such a simple filtering of the incoming scans to collect the final spectrum give a good improvement in the spectral quality, as you see to the right. Here's improvement when you also do modeling. In the figure A is we acquire data without data filtering, and we do PCA model of those data with SNV correction. First, you see the result in figure B. You can see the coding process, but it's not a very good signal to noise in that data. If we use data filtering, you see in the batch measured in figure C, and we do a PCA model of that, those data, you see a very nice score plot here that is correlated to the coding process. You see the startup phase, you see the coding phase, and when the coding is stopped, you see a plateau phase on the data here. Here are some data I've received from Sebastian Sowinski from Centronic in Germany. They have very much experience in putting NearInfrared at the end of a powder blender, a continuous powder blender, in continuous powder blending processes. So here you see a picture of the sample interface where you have powder flowing over a window, and you have inconsistent powder flow, and it's very difficult to control the measurement system. So it's a difficult application also. And Sebastian simply used also spectral filtering. It was a dual array instrument, if I recall. So you could not use the same approach that I used with Fourier transform. But here are some raw data from a process, from a powder mixing process. So what you would do is you select a wavelength that is used to indicate baseline shift. Here you see the baseline values over 900 spectra. And you put in a criteria. I only want to use these spectra with a low baseline value here at absorbance of 1300 nanometers. And if you use that approach to filter your data, you get very, very nice results when you start to monitor a process. Here, the red curve is the peak from the API. And when you start to add in the API and the continuous powder mixer, you see the concentration increase in the outlet of the mixer. And in the bottom black, you see a very stable signal of prediction of the API, and the black line is the baseline variation from the filtered data. So these are two examples of how you can filter your data and use NearInfrared for very difficult applications. So another advancement in sampling would be the use of advanced probes, and I think one of the most advanced probe is the Lighthouse probe here. The principle is that it's an automated probe that can be measuring inside the process. So you see in the upper left part of the picture here. And if you have some material that sticks on the window, you need to clean it frequently before doing measurements again. So automatically, the probe can be retracted inside an interface where vacuum and wash is done automatically. Then it can be retracted even further and you do calibration and cleanness checking with a reference inside of the interface. And you can evenly, you can take it all out and zip your whole system here. And this can be automatically. So it also is an opportunity to measure with NearInfrared in a sticky material or in an environment where you need frequent and automated cleaning and referencing. So the last technology improvement would see SIS. This is really interesting, and I'm looking forward to hear Fabien talk more about it. So I'll not spend so much time more than with advanced probe designs where you collect the backscattered spectral information from different positions. For example, from a tablet surface, you can get more information about your sample compared to a traditional reflection spectra. and I will leave that to Fabien to elaborate on this technology here. My last point, which I believe would also support more use of NearInfrared in the pharmaceutical and biopharmaceutical industry, is the regulatory guidance documents. The two first documents is, of course, the Pharmacopoeia chapter and US Pharmacopoeia. Those are the way they explain how we should set up the instruments, how we should qualify, maintain, and calibrate the instruments, and they support each other very nicely. But the two other guidelines that has been improved is the EMEA guidance from 2014 and FDA guidance from 2015. They explain in more detail what data you should use when you submit a NEAR method to the authorities, and it gives you also a good guidance how you should develop it and what you should write in your documentation. And one thing I would point out that is really interesting is the inclusion of what is called rate of change model. In the previous, when we looked, for example, on powder mixing, we always did a PLS calibration model to predict the concentration, but making a quantitative regression model requires a lot of work and model maintenance. And a good example here is Spatially and Al. There's a paper from 15, and they use a rate of change example. They use a moving F-test to determine the endpoint in a bin blender process, and they showed in the paper how you develop and you validate such a method, and this has actually been approved by the regulatory authorities. And such a method is very easy to maintain and scale up from. You can develop it during in the first phases of your production development, and then apply it in a full-scale production also. The figure to the right is a counting of what was the actual endpoint that was determined by this method over a lot of full-scale GMP batches. And you can see most of the batches, they were mixed for 45 minutes, some for 40, some for 50, and 55 minutes, a little bit more. So it actually shows that over the years, having a PAT method with, for example, NearInfrared, would give you, would show some of the variability that is necessary to get the same product quality all the time. So my take-home messages for you is that personalized medicine, for example, inkjet printed drugs, can be analyzed by NearInfrared. Continuous manufacturing with tablets, NearInfrared would really become very important for supporting such production. And raw material characterization for complex biopharmaceutical products, here NearInfrared also can be a very efficient and robust and easy way to select the right batches to get high yield all the time. And I would say that more solution to sampling challenges, you could use algorithms to filter your data, cleaning, automation, and more advanced probe designs. And then you can use NearInfrared for many more applications. And look into the regulatory guidelines. They, I think, are very comprehensive now and will support a lot of different applications of NearInfrared now. So that concludes my talk. Thank you.
Thank you very much, Dr. Skibsted. So just as a reminder to our listeners, if you have any questions for Dr. Skibsted or Dr. Chauchard, then please click on the questions tab and send them to us. We'll be asking these at the end of the presentation. So I would now like to invite the speaker for the second half of this presentation, Dr. Fabien Chauchard, to present an industry perspective. Welcome, Dr. Chauchard.
Well, hello, thank you. I would also like to thank Erik for this very interesting presentation and sharing all the experience from Novo Nordisk. Was really interesting. So thanks also for Biopharma for giving me the opportunity to present you what is SRS, which is a new technology. And today my talk will be focused on the application for bioprocess products. So just quickly to present the company. So we are a company founded 9 years ago. So we'll say more a startup company that has been bought in 2017. And we are mainly engineers specializing in optics, electronics, software. We know a bit pharma, but we are not pharmaceutics. So that's why we like to share knowledge with company like Novo Nordisk or other. And we have developed the SRS Samspec technology, and it's what I will present you today. So basically, if we look at the biotech, there's different important steps. The first one, and probably the most known one, is what we call the upstream, which is the way you prepare the cell culture. So in pharma, there's a lot of mammalian cell culture, so that are used in upstream. The upstream can also be some product coming from animal in some case in biotech. Then the second important step is the downstream, where the idea is to take the product of interest and to obtain the best purity and the best homogeneity. And then, since usually it is protein, it's difficult to store like that, so freeze drying is something that is really used a lot, and it's also an important step. So I would say more or less it's the same as what was presented Erik before. So regarding the upstream, I will not speak so much on that, first because I think that Erik presented very well what you can do with the NIR in upstream. In our case, if I present what we do quickly, that we use also quite often the Raman, because it's quite accurate, especially for measuring the nutriment. So I will not speak about the upstream part. I will more focus on the downstream and the freeze drying, and to explain you how you can use the spectroscopy, the SRS spectroscopy and multipoint spectroscopy in order to solve some problem in this field. So now I will just introduce you what is SAM-Spec technology. So just if you already look on this photo, you can see on the top of the probe there are several points to record the data. This one is a probe that is inserted into reactor, and I will explain you now how it works. So basically, Erik also speak a bit about hyperspectral technology, which is something quite new, and it comes mainly from the aeronautical industry. Basically, when you make standard image, you get an image and you try to understand the image by using the shapes that you can find in the image. For example, road will be more straight line, be clear, and then the forest will be something more black. The interest with hyperspectral imaging that for each pixel you will get a spectra. So it means that you can have the information on the pixel not by looking at the shape of the surrounding pixel, but only by using the chemical information. So you can see on the spectra if it's a spectra from a leaf, from a road, from a rock, and then based on that you can generate an image that produces information. So the previous example with the printing is a very good example where when you make the printing you can see the different points. So hyperspectral imaging is very interesting, but there are some drawbacks. The first drawback is that you still have to handle the spatial versus chemical information, and usually what is used is what is called a push broom imaging, so you need to make the sample moving. The image usually are quite big, that you have usually more than 1 giga just for a single acquisition on the sample. So it's very huge in time for calculation and analysis. And since you get an image, you have to see, for example, in the image, where is the tablet, where is the sample, and then to decide which part to use. So this takes time. However, it's quite interesting to detect contamination or homogeneity because you really have the good spatial information for that. You can also monitor change with time. For example, when you want to monitor stability, you can see where you can have some spot of instability. So I would say that there's recently a European pharmacopoeia that has been published, and it's quite interesting to read that. So, and it just provides this kind of information. On our side, what we saw is that it was still difficult to implement directly the hyperspectral camera into a reactor or close to a layer cake. So we tried to make it working a bit differently. And that's how it works. That in fact, rather than imaging the object, we use optical fiber that we put in front of the camera. The camera, inside the camera, you have what we call a slit, which is mainly the line where the light goes in, and in front of it we put this different fiber optics. So it means that you have different points and you can decide what and where you want to measure. So I can show a quick example that has been done by RCPA, where they were looking to improve the understanding of the blending. And what they have done is that they took their blender and they put several points to measure. So you can see here on this drawing you have like 10 points with optical fiber, and what they can get is that during the mixing, they can measure simultaneously all the points, and so they can really generate some kind of modeling to see how well the API is being blended in real time. So you can even optimize your process by deciding which plate to use, what is the best shape of the reactor. So it's really interesting, and it's really based on this multi-point approach, and something we use often. So now I will just go back to the spatially resolved spectroscopy to explain you how it works. So I suppose that probably all of you know the sky, and that the sky is changing in color. So usually when an object is changing in color, what you believe that something is changing. For example, if you take an apple that is really, let's say, yellow, and then it's getting red, you will say, well, no, there's probably more sugar inside because something chemically has changed inside. However, if you look at the sky, nothing is changing. Still the same sky at midday or at sunset. What is really changing, in fact, is how the light is being scattered into the product, and it's more due to a physical phenomenon. So it's where it's important to see the 2 points, which are the physical and the chemical part. If we look at 12 o'clock, in fact, the path length into the sky is very short compared to the density of the sky. So mainly the blue is scattered. So that's why we can see it, but it's just scattered in order to get some absorption. So we can see the absorption from that, and we can see the blue. However, at sunset, the path length is much more longer. So what is happening is that the blue is completely scattered before getting back to you, because there's many times more scattering even. So what you see, in fact, is just the remaining color, which are the red. So you can see on this simple example that sometimes it's better to, in order to know very accurately a product, to measure at different points, because like that you can know the chemical information and the physical information due to the scattering. And so that's how it works. For example, if you want to work with small and large particle, what we do is that we make measurement at different points from an illumination source. And what will happen is that depending on the density of the particle and the mean particle size, we will get more reflection or less reflection, and we will see this shape of reflection. And based on that, we can analyze both the spectral one for the chemical information, but also the spatial one in order to get the information. To give you another example, it's just like when you have a motorcycle in front of you on the road, and if there's fog, if the fog is more or less dense, you will see a bright light more or less big, and it's just due to the scattering of the light into the fog. So here's a more detailed explanation on how it works, and it's also a study that has been done by RCPA, where you can see that by just taking fiber at larger distance, also you can increase up by 3 the distance measured into the sample and also the volume, which is something quite important, especially if you are analyzing product that will be delivered to customers. It's important to have a very large investigation point. And it's also something that has been pointed in the pharmacopy, where they really also say that even for imaging, the sample surface representation is something important, because usually hyperspectral provide you only the surface of your product, while what is important is also the subsurface information. So the spatially offset measurement is really interesting because you can capture this information below. And it's the same for the sample preparation. It's also important to prepare this sample according to what is designed into your product. So now I will give you some example of application on how to use the multipoint and how to use the spatially resolve in the biotech. So the first one is a typical filling line, and it's also something interesting because it can be also a continuous filling line, because now in biotech there are also continuous processing and continuous downstream. So in this case, you have different steps and different parts. You have the feeding of the reactor, then you have the reactor itself that is usually under agitation in order to keep something homogeneous. And then you have the filling of the vial, and usually this is a closed loop where if there's no filling, then it goes back into the reactor. So in fact, there's many critical parameters, such as the concentration of the product, the physical property, such as the homogeneity, and sometimes the turbidity when you have some kind of mixing, like for example lipid suspension. And there's also risk of sedimentation, risk of crystallization or falling into the pipe, so several critical points. Now, if we look at where we have to measure, usually it's difficult to say that there's only a single point that is critical. Usually there are several points, and if you use a standard spectrometer with just one channel, something that can be tricky to handle. So what is good is that if you have a multi-point approach, you can decide to measure at several different points and get some kind of real-time imaging of your process that is running continuously. So typically here it's an example of system we have used. So here it's a multi-channel system, and you have different way of analyzing your process. You can use a standard probe that you connect into the reactor, but you can also use a flow cell, or you can also use, I would say, a transmission probe. Then regarding the communication with the process, usually what we can find in the pharma is the OPC UA that allow communication with different kind of software to monitor the system. So here what is interesting is that what we have tried to use is also something that is being a bit new on the market, that we're speaking about NIR spectroscopy, but now there's also some spectrometers that are quite wide. It means that you can measure from the UV up to 1000, which is in the NIR. So it's quite interesting for the downstream, because in the UV you can find the protein, some API, some even petrochemical. You can also predict DCO, DBO, or analyze nanoparticle. In the visible, you have some important information also regarding to some component, for example, carotene, color, and even if you are looking to hemoglobin, it's a very interesting part because you can see the oxygenated hemoglobin or deoxygenated. Then if you switch to the NIR, that will start at 750. You have, of course, first the turbidity, which is a standard where you have to measure the turbidity at 830 or 860. And then you have even the absorption band of water and the absorption band of glucose and even lipids. So in a single shot, you can have a quite good view of what you are working on. So here in this case, we connect the spectra at different points, and what can be done then, once you connect that, you apply directly a chemometrics model, because I think that Erik has explained that very well, and he showed all the different methods that you can use, such as classification, filtering, and then the PLS model to predict, for example, the purity level. So all this must be implemented into the unit. And then in order to work with closed loop, you have to use different kind of software. So here I just put an example of software called SynthQ, but the idea is that this software will get the information from the analyzer, and then it will be able to act on the production tool to, for example, make the agitation higher, or just provide more feeding, or to change even the feeding into the reactor. So it's something very important to have, of course, a good measuring tool, but also a good strategy for making the automation. Then I will switch now to the freeze drying. So the freeze drying is something very critical. What is usually most important is also to validate the moisture concentration and to check that the moisture is good. So there's two steps. There's the first step where you want to improve the freeze drying itself, and then there's the second step where you want to check the lyo cake before selling it. I will provide you an example that has been presented by a company in Spain called Ray Joffre with Bonsai, and they were, for example, using the probe inside the freeze dryer just to validate the cake and the density of the cake. We can go even more deeper, where we want to get some kind of mapping of what is happening into the freeze dryer. And in this case, of course, you cannot decide to measure just one lyo cake, because the freeze drying is something quite complex, and depending where you are measuring on the plate, you may have different things. So what is important is to be able to have some kind of mapping, and of course, in this case, it's important to have a multi-point approach to be able to measure at different points into the freeze dryer. And it's really that the idea is to be able to validate then the endpoint, but also to have a good knowledge on the process, to know how to generate the best ice crystal, and how to get efficient process and fast process to remove the water. So this is also taken from another study from Ari Kopinen in Finland, and they have used a multi-point hyperspectral camera to measure at different points on the lyo cake during the freeze dryer. So they have been able to see dynamically during the freeze drying process how the water is getting out, but also what was the ice growing if you was having a small crystal or big crystal. So it's very interesting approach. And you can see on this next chart that they have been able to design, I would say, more standard process, but also more aggressive process that can go faster, and in order to reduce the time, and also to generate some very specific crystals. So you have the reference in my presentation, but you can really read this paper. It's very interesting. Now I will switch to more the analysis of the lyo cake once it is finished. And usually something very important before selling the product, to be sure that the residual moisture is, for example, below 1%, to be sure that the product is stable. And it's also something where the pharmacopy really give specific guidelines to say that it's a critical parameter and that it must be measured. So basically, since we have the SRS, we can apply a different way for measuring the lyo cake. We can measure from the bottom with different optical fiber the lyo cake through the vial, or we can also decide even to measure from the bottom and from the side. Why is it important to measure so many points? In fact, the cake is not completely homogeneous. Even the way the water gets out, sometimes you can have different water content into the cakes. So that's why it's very important first to have a big volume analyzed, to be able to have a subsurface information and not only the water content from the surface. And also, if you have several points, you can also see the cake, for example, the cake density, if the cake is dense or not dense, and you can even see if you have some melt, which can be a problem into the cake, or even if you have some kind contamination, for example, a part of plastic that can fall into the vial. So this is an example of setup. So here you can see on the top measurement from the side where you have the sample moving, or you can have also a measurement from the bottom, or you can even have measurement from different sides. So what is interesting that the probe is very small, so compared to a camera, it's easy to install on production line, and it's also something flexible that you can easily use for the lab. We also use spectral filtering in order to generate automatic trigger, because you can see on the graph that these are the spectra taken when the vial is going in front of the probe, so you can clearly see a higher intensity when the vial is exactly in front of the probe. So this is an example for the moisture measurement. So here the prediction is done in very fast way. Within 3 milliseconds you can get the information, and you can see that you can measure easily up to 0.2% of residual moisture into the product, and this is mainly due because water is a very strong absorption band, so you can go to quite low concentration compared to other chemical constituents. So usually the calibration is performed between 0 and 3%. And the idea, of course, is to get for the calibration step different concentration, and then during production validate that all the production is below the critical limit. So this is the example. You will specify the bandwidth, and then you will check that everything is good and homogeneous, and then you can rely and reject in real time the layer. This one is another approach to try to detect contamination. So here it's more based on chemometrics, that when you get different spectra, you can try to classify these different spectra. So this is an example. Since we are measuring with multipoint, when the sample is passing in front of the probe, we can have up to 300 measuring points. So it means lot of spectra. And so we can try to look at all these spectra to see if all the spectra look like the same or if there's different spectra. So you can see that, for example, with this PCN, then you can detect some abnormal spectra, and you can try to see if they are related more to glass or to other products, or if it is a more physical problem, for example, a crack into the layer cake. The other approach also is to use what we call the homogeneity index. So the homogeneity index is basically that you just compare the standard deviation between the spectra. If all the spectra are the same, you are supposed to get some kind of spectra flat. If then you have non-homogeneous product, then you will see on this standard deviation some peaks that are related to the product that is not homogeneous. So here it's an example that has been done on tablet, real-time sorting of tablet, and here in this case, in fact, the tablet was not homogeneous, and this peak were exactly the peak from the API, so it was possible to detect that there was a problem of uniformity on the tablet. Here's another example for measuring not only the API but also physical information. So here the hardness of the tablet was calculated by looking at the difference between the SRS signal, which are the different points from the light source, and based on that it was very easy to separate the different hardness. So the hardness of the tablet will act more or less just as the sky getting blue or red, that if it's getting more or less dense, you will have more or less scattering, and it's what we're able to see with the different measuring points. So now we switch to the conclusion. So the conclusion on multipoint and SRS is that it provides a new way of analyzing the product and developing strategy to control the process. In biotech, you can have very fast technique based on the imaging sensor that is compatible with high throughput and multi-lane machine, because most of the packaging line they use several lanes. So since you are able to use several probes, it's more easy to put on production machine. The Second point is that the multipoint technique is ideal for multi-batch, line, continuous, or even continuous process with powder. Since there's different process steps, you can have a look at all the process steps with the same analyzer, and like that you are able to get a real-time image of your continuous process and to act in real time also on the process in order to keep it on track. The third point, which is the most important regarding the interest of SRS, is that most products in pharma are complex. They can be emulsion, they can be tablet, they can be powder. So they all have physical parameter and chemical parameter that are both important for delivering the drug, because the density will play a role on the dissolution, and the API, of course, will also have a high impact on how the drug is going to act. So that's why it's interesting that it's a technology that is able to provide the two critical parameters, physical and chemical, to validate the product. So that's all for my talk. So I will be happy to answer questions, so don't hesitate. We're listening to you now.
Thank you very much, Dr. Chauchard. So at this point, I would like to begin the question and answer session. And as mentioned previously, if you've got any questions for us, please click on the question tab and send them through to us. So our first question is directed towards Dr. Skibsted. What is the most critical part when developing an NIR-based method for screening of soy hydrolysis?
Yes, it's the same problem for any complex raw material and cultivation or fermentations. It is to look on your cultivation data and to find objective way to say it's a high yield or a low yield, because it can be very difficult to analyze the drug product inside the bioreactor, and that's actually the data from where you want to evaluate. So in our case, we did cultivation in small scale for 18 days. And during 18 days, we took out like 5 samples for the titer. But it's which should you use all of the 5 data points or only a few of them? The first data points, the first days, there will not be much difference. In the middle of the cultivation, there will be much more difference between the different batch runs. But we also found that if we support the data analysis with information about cell density, about the viability, you get a much better insight in the performance of the soil. And that was the reason we took all the available data and the PCA model. It's a filtering technique to look on information versus noise. So my advice is to spend a lot of time on analyzing your cultivation data with the chemometrics. It's not so difficult to do near analysis in a robust, reproducible way, but it's very difficult to you should put your energy on your reference cultivations.
Great. And the next question is for Dr. Chauchard. What is the accuracy of the system for measuring moisture in Lyell, and how many vials can be analyzed per hour?
So the track accuracy can be also related to the speed that you are looking for, but the standard accuracy is around 0.2%. And of course, the speed is an important parameter, because depending on the speed, you can have much more point measure, and if you have more point measure, of course, you can decrease the error. But usually, let's say that we try to analyze around 100,000 vial per hour, which is usually compatible when you unload the freeze dryer, because usually it's where you want to check that the moisture is below the critical concentration.
Great. Next question for Dr. Skibsted. What could be the next technology leaps within NIR technology?
Yeah, it's a good question, and maybe I think Fabien and his colleagues is maybe working on that. I think more multi-point technology, and ways to improve the sensitivity of the NearInfrared. NearInfrared is sometimes not chosen because the sensitivity is lower compared to other techniques, for instance, infrared. But I would say any advancement in improving the sensitivity of the technology, maybe in more advanced probe designs, could really make some differences. I think the sampling problems, there are many great solutions for that. So I think improvement of sensitivity.
Great. Dr. Chauchard, did you have anything to add to that at all?
Well, I would say it's quite agree with Erik. That's true that NIR is quite good technique, but usually the people they don't take enough time to understand the sampling. And I think that in Erik's presentation we will really see that the sampling, where to measure, how to measure, and take some time also on the filtering technique can really increase the accuracy and the performance. So I think it's also, if we compare with Raman is interesting, but it's much more complex to decide how to measure and where to measure. NIR, it's really flexible. You can measure in transmission, backscattering. It's not so complicated to make new way of making the measurement during the process. So I'm quite agree with what Erik just said.
Great. And the final question for Dr. Chauchard is, for downstream process, would you recommend to use UV NIR or Raman spectroscopy?
Well, it's a bit related with what just Erik said. That so that depending on the drug you are using, the both can provide different sensitivity. Sometimes the Raman is better, and sometimes the UV is better. But UV and IR it provides a quite interesting approach, because of course you can have different way of measuring different sampling technique, just as we say. And the UV is very sensitive to some molecule. In some case, for example, for controlling wastewater in industry, we have been able even to detect ppm of API into the water, and being able to say that there was still remaining API into the water, and it was not possible to throw away this water. So that's true, the UV can be quite sensitive. On the other hand, the Raman sometimes can be easier to understand, because there are some amine absorption bands. So it's usually we always propose to the people where we are working with to make some tests to check which one works the best.
Okay, well, unfortunately that's all the questions that we have today. I would like to take this opportunity to thank our presenters, Dr. Erik Skibsted and Dr. Fabien Chauchard, for sharing their knowledge with us. Audiences can view this webinar on demand by visiting biopharma-asia.com/webinars. Thank you, everyone.
Thank you.
Thank you.